Welcome to Pharmaceutical Calculations, a Conceptual Approach, Lesson 25, Fluids and Electrolytes, Part 2, Osmotic Diuretics. Today's objectives are to describe the properties of osmotic diuretics and describe or discuss their similarities and differences from plasma expanders. Today's lecture, unlike the very long lecture of Lesson 24, is very short but a fascinating one. Now, we need to cover osmotic diuretics in the chapter of fluids and electrolytes because osmotic diuretics are commonly used to facilitate water removal from our body. So let us now go through some of their indications. Osmotic diuretics are used to treat conditions that are associated with decreased urine volume or accumulated fluid in the interstitial spaces of organs such as the brain. They are commonly used to reduce intracranial pressure, to reduce intraocular pressure before surgery for glaucoma, to increase urine flow in acute renal failure by diuresis, and to accelerate renal excretion of water-soluble toxins. The most commonly used diuretic is mannitol. Now, what about their mechanism of action? These molecules are so unique, they are so fascinating, that essentially all the effects in our body is solely due to their physicochemical properties. They are totally devoid of biological activity. They do not bind to any receptors, they do not bind to any enzymes, they don't bind to any proteins. They essentially work by increasing the osmolarity of plasma, obviously because they are polar and water soluble. Their polarity prevents them to be distributed inside the cells because they are totally devoid of hydrophobicity. They cannot cross plasma membranes by diffusion. And because of their small size, their low molecular weight, they are freely filtered into the urine, and therefore they drag the sorbed water into the urine. Okay, so this is their essential difference from plasma expanders. If you recall from lesson 24, plasma expanders are also polar. They are also very water soluble. So they sorb water into the plasma. But because of their high molecular weight, they cannot be filtered into the urine. They stay in the plasma and they increase the plasma volume. This is the difference between plasma expanders and osmotic diuretics. Osmotic diuretics are very low molecular weight. They are filtered freely into the urine because of the high osmolarity of the urine now, since the solute concentration is increased in the urine. They are able to eliminate plasma volume by increasing the rate of urination. So this is called diuresis, okay? All of these important points are mentioned nicely in this paragraph over here. Again, there are essential difference from plasma expanders is the low molecular weight. Because of their low molecular weight, they are able to be freely filtered into the renal tubules, and they drag the water that they sorbed from the plasma in the urine, and they increase the rate of urination, thereby increase the rate of water removal from our body. Now, what are the desired biophysical properties of osmotic diuretics? And I was looking for a word that emphasizes the fact that osmotic diuretics are totally devoid of biological activity. So let's go through some of these properties. First, they have a very high polarity. They're very polar, they're very water soluble, right? They have a very small molecular weight and therefore can be filtered through glomerulus. They remain in the extracellular space. They cannot cross plasma membrane. They cannot get inside the cells because they have no hydrophobicity and they are very polar, okay? Also, they don't get inside the cells because they are not substrates of any protein transporters. So passive diffusion is not a choice here because they are very polar. And at the same time, because they are not substrates of any protein transporters, they cannot actively be transported inside the cells. They do not cross the blood-brain barrier. They are not reabsorbed, obviously here, back into the blood, right? Back in the blood, in the renal tubules. This is what we mean here. Because if the osmotic diuretics, if they were reabsorbed back into the blood, they wouldn't be able to keep the osmolarity of the urine high, and there wouldn't be any water removal. So this is very important. They are not actively secreted into the urine. And obviously, I'm not certain how important is this to their action here, but obviously the fact that they are freely filtered in the urine is much faster, and they drag water with it. They are not metabolized. That's important because they're not losing their activity. If they were metabolized, they would lose their activity. They are freely filtered through the glomerulus, we have just described that, and they are primarily excreted in the urine as the mode of elimination from our body. Of course, this is related to that, and of course to that. Okay, remember that reabsorption back into the blood requires hydrophobicity. This is not an active reabsorption. Okay, since they are very polar, they remain into the urine and they drag water with it. And the last one I've mentioned so many times. Let us now go through some of the calculations involved with osmotic diuretics. So here in example 10.5, we have a 154-pound male adult that is in the emergency department with acute renal failure. He's to be infused at this dose, 200 milligrams per kilogram, over 10 minutes. That's what I meant, very fast infusion with mannitol. The mannitol solution available is 10% weight per volume. You need to calculate the rate of infusion in ml per minute. So here we have three small steps. The first step involves calculating the amount of mannitol based on this dose here. So we need to convert the pounds into kilogram and then calculate the milligrams of mannitol. And then use the milligrams of mannitol to decide how much volume of this solution needs to be infused, and then divide by the time to calculate finally the infusion rate. Okay, so let us go step by step. The individual is 70 kilogram, multiply by 200 milligrams per kilogram, and you get 14 grams of mannitol. Divide by the concentration of mannitol solution, and you need to infuse 140 ml of mannitol solution. Divide by the time of infusion, and you get the rate of infusion being 14 ml per minute. This is a very fast rate. Okay. Finally, we are in the practice exercises here. These are very, very informative and nice exercises for you. So here in 10.4, we have two molecules, mannitol, which is an osmotic diuretic, and cholesterol. And I'm asking you if cholesterol could be an osmotic diuretic. So all you have to do here is to perhaps see the structure of cholesterol and predict some of the properties. Is it polar? Is it hydrophobic? Is it water soluble? Is it high molecular weight? Can it go through plasma membranes, right? And figure out whether cholesterol could be an osmotic diuretic. In exercise 10.11, you need to mark the statements as true or false. Perhaps you can go through one or two. Let's go through A. Plasma expanders are eliminated by the kidney. Now we know that plasma expanders, they are high molecular weight and they are not. Osmotic diuretics are hydrophobic compounds. Obviously, this is also false. We know that in order for them to absorb water, they must be water soluble and they must be polar. Otherwise, they would partition inside the cells, right? So you may read the remaining statements yourself. And finally, 10.26, a very nice exercise, very informative, and of course, you need to practice your calculations here. You are given the dose as 40 grams per square meter, so you're going to have to use the BSA equation here, the body surface area equation, to normalize the amount to calculate the amount of mannitol as the dose that is needed to be infused. And you have the answers here to make sure that you have solved the problem correctly. And at this point, I would like to thank you for your attention. Until next time.